you're from the area. But even if you have done that, you probably may not know what's going on here. This place is changing very fast, and that's part of the reason I came here a few years ago. Last week, the U.S. News and World Report came out with their rankings of undergraduate national institutions, and we, for the first time, were in the top 50. And that is impressive, of course, because there's a lot of competition up there. But at the same time, when you consider when I came here 3 years ago, we were 69. We've come up 20 places in 3 years. Now, it's not my responsibility at all. This is just the momentum this place has. If you go back another 5 years, we weren't even in the top 100. So why is Northeastern rocketing up in the undergraduate rankings? And the answer is co-op. So those of you who've interacted with us this way, and I think some of the companies here have had our co-op students, probably understand this very well. But the co-op model, basically, our students go out and work for 6-month periods, either 2 or 3 times during their degree. And this is a fantastic model of education in my mind. Obviously, it helps you find a job. It helps you figure out what you want to do. At the same time, it's a great way to illuminate your experience in the ivory tower. I spent 2/3 of my career outside academia. My first job was at Bell Laboratories in the 80s for 11 years, and so I've worked in industry and national labs. And I really think that this coupling of industry and academic environment is really important for both sides. So that's what we're selling to our students. That's why we're attracting such outstanding undergraduates. And for us, it's an exciting challenge to be attracting the best students in the country. They don't just come here because they want to get a job. They come here because they realize that even if they want to go on to graduate school, this is a fantastic way to develop their passion for life, figure out what they're really good at, learn about themselves at the same time. So that's very exciting, and that's going to continue. I don't see any evidence that we're going to slow down in terms of attracting outstanding students. In parallel with that, though, we're also developing our research. Obviously, that's a critical, an undergraduate education in a research university. Research is critical, research experience and so on. So our faculty are leading in this area. We're hiring a lot of faculty. It's one of the reasons I came here. We have 6 departments in the College of Science, and in those 6 departments in the last 3 years, we've hired almost more than 30 people, faculty. And we plan to hire more, probably another 20 or so in the next couple of years. That's very exciting, and we're being very strategic about that. So we're picking, together with the faculty, we're picking areas where we can really take some leadership and partner with others. So in general, the area of drug discovery, delivery, and diagnostics is one of those areas where we're already strong, and we're making strategic hires and picking areas. So obviously the CTNI fits into this. There are a lot of activities. We have medicinal chemistry. We have an emphasis on neglected diseases. We have behavioral neuroscience. We have regenerative biology. And these things are coupling together into a real powerhouse. And we're really interested in extensive partnering with people from the region. And we're strong in some areas, but not in others, and we can work together. So I'm delighted to see you guys here today, and hope that not only will you take advantage of the imaging facilities, but also see some other opportunities perhaps to extend collaborations with Northeastern. So with that, I'd like to welcome you again. Thank you for coming, and hope that you have a very good day. Hand back to Mark.
Thanks, Dean. Before I hand it over to the morning's moderator, I just want to also acknowledge and make sure the Dean is aware of the fact that I'm an alum of Northeastern, although a late one through business school. And actually, my daughter's graduating with her doctorate in physical therapy this year. So, yeah, I get those envelopes all the time, don't worry. Last thing, we're small right now. Certainly, there are a lot more people on their way. I want to encourage everybody, and I think Peter does too, to make this a very interactive and engaging forum. It's small, it's intimate, so I suggest everybody get involved. So with that, I want to introduce Mike Luther to moderate and guide us through the morning. Mike is the Senior Corporate VP of Discovery at Charles River Labs, has held a number of senior leadership roles at major pharmaceutical companies, and has a great deal of drug discovery and development experience. With that, where's Mike? Oh, there you are, Mike. It's all yours.
Good morning. I get the pleasure of moderating this morning's session, and before we get into that first session, I just wanted to take a few minutes to talk about the paradigm and the call it the hurdles we face in sort of translational drug discovery. It's an easy term to say, a very difficult one to practice. Having spent 25 years in the biopharmaceutical industry, and most of that, almost all of that in the drug discovery area, I've seen, from the early 80s through today, the accelerating amount of R&D dollars that have gone into drug discovery, the creation of a whole biotech industry that we've seen, and at the same time, as many of us are aware, that these R&D dollars have not translated into new chemical entities or new therapeutics, and there's still a tremendous unmet medical need out there, especially as we have an aging population. At no point in time, I believe, have we had the opportunity to see the convergence of technology in the life sciences, the human genome, a lot of the new, call it detection technologies. We're going to talk a lot about imaging today. We also have, in terms of IT and informatics, unbelievable tools that are coming together. The question is, how do we apply these tools in our toolbox to ensure that we actually translate from basic drug discoveries into the clinic with a higher probability of success so that we can see this investment going through? A lot of the issues have been is that, and I can say that I've cured a lot of rats and mice of diabetes and cancer and arthritis, and many times those have not translated into the clinic as successful new entities that actually benefit patients. We all know that a lot of our preclinical models and approaches are not predictive of the human condition. And so that's that's the challenge we have. Many of the tools I look at it today is helping us better understand pathophysiology in our model systems and how does that pathophysiology relate to the human condition. And I think that is a challenge that we all face today. And as we look at that, how do we add pixels in terms of how do we build a high resolution picture that gives us greater confidence of what we're looking at is actually relevant to the human condition. And I think today, we have the basics tools. The challenges are actually how do we use those tools better. If I look back across the last, say, 25 years, there have been these, call it scientific paradigms that we said we're going to change the way we think about drug discovery, and these are going to be the solutions that we had. I think about early on, it was the intent of molecular biology and gene expression, and then there was structure-based drug design, and then there was combinatorial chemistry, and then that combinatorial chemistry and molecular biology coming together and saying we're going to do high-throughput screening so we can screen the entire human genome against these, whether it's a compound or an antibody library. The point was we took sort of this reductionist approach to drug discovery, and we know that the reductionist approach to drug discovery has failed. We have to be able to look at the system and understand the system in toto. And that, I think, is, if you think about what we're going to talk about today, is really where we're going to focus a lot of efforts. How do we actually systematically look at these systems? And obviously a big focus today is on CNS and imaging. And imaging is a highly translatable tool. And as we see it, it moves further and further back in terms of all the way from basic research being used in drug discovery and then discovery into that clinic. And so I look forward to hearing many of the speakers today and, like I said, have the pleasure of moderating this morning's session. So with that, I'd like to introduce our first speaker, who is Dr. Peter Lansbury. Peter has a distinguished career. He has a Ph.D. in organic chemistry from Harvard. And he worked for E.J. Corey, and so anybody who, at least when I hear that name, I think about many of the medicinal chemists that I have had the pleasure to work with. I mean, hold EJ in very high esteem. He was an assistant professor of chemistry at MIT and was promoted to associate professor in '93. And then he's moved in '96 to his current role where he's the professor of neurology at Harvard Medical School, and where he founded the Laboratory of Drug Discovery and Neurodegeneration. And so with that, I'd like to welcome Peter and look forward to his talk.
So now we really want to keep it informal, because only the true believers are here. So before I start, I want to say, because I'm going to give a talk that could be interpreted as negative, as a little pessimistic. I would call it realistic. But I just want to say that I think this is, for me, it's really like a sacred quest, trying to break this logjam and get at neurodegeneration in my case, Parkinson's disease especially. But I want to present a talk that deals with sort of the realities of the problem right now, and some of the ways that I think that collectively people can make changes. So that's I just want to say I admire all you guys for being interested in this. It's really, really, really important, as I'll talk about a little later. My background is not in pharma, so I was really a very basic academic scientist who was working on sort of the biochemistry and biophysics of protein deposition diseases. And then I had a midlife crisis where I didn't buy a car or anything, but I decided to do something practical with my life. And the first thing I really focused on for maybe 5 years was thinking about how academics can work with pharma and maybe even do some drug discovery in an academic setting. And I also spent a lot of time railing against the pharmaceutical industry for being only interested in profit. And then I realized that was the whole point of the pharmaceutical industry. So the last 10 years I've been involved in a couple biotech companies. I founded one, went on an extended leave for about 5 or 6 years. That company was sold to AstraZeneca about 2 years ago, and now I'm involved with another one. I want to tell you those just stories because they illustrate some of the issues and problems later on. But first off, I sort of want to introduce the talk with kind of this thing I stole from an advertising book that my wife had me read. Thinking inside the box, and of course the box here is capitalism, really. It's that there's so much risk inherent in this problem, and we can talk about why, that something has to be done to reduce the risk or to change the people who are paying for the research. I think maybe in the afternoon there'll be some discussion of that in the afternoon keynote. But I think that's the real challenge, is using creativity and harnessing this creativity and this blue skies thinking, but really putting it into a context of, if the risk is too high, no matter, if you have, the next cure for Alzheimer's disease in a bottle, people are not going to try, especially in the current climate. And I'll talk a little bit about why that's true. So, we can look at statistics, population statistics, and the latest I heard, and I'm going to paraphrase it, so it's probably wrong in detail, but it's right in the general idea, is that in this country, in the next 30 or 40 years or so, there's going to be a doubling of the number of people over 80 and no change in the number of people under 50. That's scary, right, from a healthcare perspective. And also, if you look at the chunk of the GDP that Alzheimer's disease is going to take, it actually becomes a significant chunk, like in 2030, like 10, 12%. Because we're just going to have this increasingly aging population. There's absolutely nothing for these patients, but they're going to live longer because they're going to be getting good care in other areas. And so, this is a huge, I would argue it's almost like it's a national security issue. And I think the NIH recognizes that, but they don't, asking someone in government to look 30 years into the future and pre-plan for that is crazy. They're doing it in Japan, but of course they're not doing it here. So their commitment to this area has been really minor. I went to this summit at NIH, and they trotted in Kathleen Sebelius, to announce this big new initiative in Alzheimer's disease. It was $160 million, which is a joke. I mean, that's 2 clinical trials, And so they haven't picked up the slack, although they recognize it, and I think, it may be just a matter of time. Meanwhile, pharma's just getting out. And so you may know Novartis fired their whole neuroscience group in Basel about 2 years ago. A really excellent group with a long history, great science. AstraZeneca fired about 400 people in Sweden and replaced them with about 40 people here in Cambridge. And I don't understand, they're sort of outsourcing everything, doing things the same way. So I think it's a commitment to losing less money, basically. I don't think it's really a commitment to doing things differently. Pfizer fired 2/3 of their CNS group when they were down in Groton. They brought the rest up here. So it's a real disaster. There's really a lot of people getting out and very few people going in, with the exception of multiple sclerosis, and we can talk about that, why that is later on. This is just to remind everybody that it takes a long time to get a drug. This is probably too short. But as we go from the initial discoveries to lead compounds, and then through phase 1, where you have a compound through phase 1, now the chances that you'll get an approved drug, as you continue through this pathway, get greater and greater, and so the value of the drug becomes greater until the drug is introduced. And the costs increase, of course, a huge amount. In fact, this is the point not only where the chance of success starts to register, but also where the costs really explode when you have to, when you go into human trials. So I want to come back to that in a second because Alzheimer's provides some unique problems. So the issue basically is that everyone who's investing in AD research right now, since the government isn't really doing it, has a profit motive in mind. So they have to consider the risks, and the risks are just way too high. The number one risk is that it might be untreatable. We don't like to say that, but that's a risk. And so once a disease is shown to be treatable, like MS, everybody is willing to rush in. The risk just drops a lot. This is something that's never discussed. The other thing, which has been discussed a lot recently in the press, popular press, is that maybe when we diagnose the disease, it's already too late to treat it, right? And maybe we have to diagnose much earlier. Now, my opinion of that is that there's no evidence that says that that's the case. It sounds like vaguely like a cancer argument. So people intellectually, it's pleasing, that story, they like the story. People who are developing imaging agents that detect early changes like the story because it promotes funding for their research. But there's no evidence that that's the case. And if you think about that in practical terms, you're going to be treating people that aren't really sick, and so you have to change the definition of sick. It's a problem. I mean, it has been done with cholesterol, because people with high cholesterol aren't sick. Right? We're just treating them to prevent them from becoming sick. So that's a real problem. And this is a study that was done, published in the New England Journal last year. These are patients who have dominantly inherited forms of Alzheimer's disease, which are called mutations in the presenilins or amyloid precursor protein. It's a very small slice of Alzheimer's disease. But in these patients, one can follow these patients and one can estimate, based on a mutation, the time to first diagnosis for these patients. And so you can piece together this information and get sort of a longitudinal view of the changes that occur in the disease. And you can see there are a lot of changes that occur even 10 years before diagnosis with the disease. And those changes include the amount of A beta in the brain. This is detected by a PET scanning imaging agent. They also have changes in glucose metabolism in the brain, also a PET scanning agent. CSF tau in the CSF, A beta in the CSF are changed. And this is, of course, this is a COG1 cognitive score. Hippocampal volume doesn't look like a good marker to me. Actually, A beta here doesn't look like a very good marker to me either. Because once you are diagnosed, it's over. I could argue from this, and also based on the scientific evidence, that A beta deposition might be a protective mechanism. And when that mechanism fails, you start to exhibit symptoms. right? And, there's no, I don't think there's any science that would argue against that. But the next problem is that because the disease progresses so slowly and it's so variable, right? An Alzheimer's patient could be great one day, bad the next, right? And probably a lot of you have had the experience I have with my father now, you have to run a trial that is about 18 months long to see a change in the rate of progression. And that trial has to have a lot of patients. So some of the trials that have been run recently have 1,500 patients. That's a huge effort in like 4 continents, $100 million minimum for a trial like that. And a long time waiting, right? You add that to the end of the typical development, right, pathway. Let's say if you're making a cholesterol lowering drug, the phase 3 is, might require a lot of patients, but you make a very acute measurement, it's over. This is 3 extra years. That means 3 fewer years of patent protection at the end. So the compound that you developed back at the beginning has 3 fewer years on the market. And 3 fewer, that's enough to really knock it off. So despite the fact, everyone says, well, gee, Parkinson's is so profitable, or Alzheimer's, everyone should be working on it. No, that's really, actually, it's not the case. And the risk is so great that it doesn't justify the investment. And so really the risks here in these early phases, not really phase 1, but especially in phase 2, are really, the risk reduction of phase 2 is less than you would get in a typical study. Yeah.
Peter, I'll try to
Sure.
be interactive. With regards to patent life, do you think that the government, the patent office, should allow, or provide some concessions to companies?
Yeah, I think that now you're getting me off. I'm going to go on. The Orphan Drug Act, right, has been incredibly impactful on drug discovery. That says when you're doing research on a drug for fewer than 200,000 U.S. patients, you get special concessions along the way, tax breaks, research refunds, and also you get 7 years of guaranteed exclusivity on the market, which is more than you would get with a normal compound, given this elongated phasing. So now there's more research going into orphan diseases than ever before. There are a lot of successes, and people are getting out of Alzheimer's. It makes no sense, because what they've demonstrated is if you understand the problem and reward people, they are going to go after these diseases. So something like that could have a huge effect on Alzheimer's research. And it wouldn't require a spend at all, really. I mean, there'd be tax breaks maybe involved, but the key thing is exclusivity in the market. Finally, I think this is the biggest problem that no one really admits, and that is that I don't think Alzheimer's is a single entity. I'm not a neurologist, I'm an organic chemist, but if you talk to clinicians, the clinical heterogeneity of this disease is enormous. They know it's not one thing. Alzheimer made it be one thing by saying that when he took 6 patients and looked at their brain, they had amyloid plaque. So he made amyloid plaque be part of the disease. It's not a biological truth. It's just that he made that correlation and defined it that way. But I think it's very unlikely to be a single entity. And I think people in the field want it so badly to be, and they focused on amyloid because that, by definition, is sort of a single, right? Makes it a single entity because Alzheimer defined it that way. They focus on it because they think that if it was a bunch of little entities, then pharma would be even less likely to go for it because the market at the end would be less, the pot of gold. But the fact is that if it was a bunch of diseases of 150,000 patients, pharma in this climate would be much more likely to go after it because they could take advantage of orphan drug rules. And if they had a better understanding of subgroups of patients that were being driven by the same underlying biology, they could focus their clinical trials. The clinical trials could be much smaller. The reason the clinical trials have to be so big is because the variability is huge. There's so many people who don't respond at all, and then there's people, it's all over the place. And even in the placebo group, they're all over the place. And so this is a really important point, and it's something that I really want to try to spend the next couple of years on at least, is this idea of trying to reduce the heterogeneity and understand underlying biology and then treat the underlying biology. The reason it's defined this way, of course, is because the symptoms also can be sort of grouped, even though that is extremely difficult to write. As I said, there's a lot of heterogeneity. But since we're not treating symptoms, we're treating underlying cause here when we're looking for a disease-modifying drug, at least. It's not a useful definition. Neither is the definition of Parkinson's disease. And that's a definition that was come up with by a guy who was sitting in a park in London watching people walk by, and he observed the shuffle and the bradykinesia and the tremor. And those are a collection of symptoms that not all people with Parkinson's have, and that could come from a lot of different places. So I think the problem with neurology is that it's defined by symptom presentation, right? And if you're trying to design drugs, you want to design drugs that are focused on underlying biology. So neurology has to undergo sort of the revolution that oncology has, where you don't treat a tumor based on where it's at, you treat a tumor based on what that tumor is and what markers it has. Here's just one example of this heterogeneity. Even the pathologists all recognize it, right? The guys who are guilty of propagating the amyloid plaque actually recognize that it's probably a bunch of different things. So I want to talk a little. Mark, you're going to interrupt me and tell me when I have 10 minutes left.
Oh, Mike is.
5 minutes? Oh, Mike. Yeah. Okay. All right. I didn't know if Mike was willing to be rude, but please do. So maybe you know this story, but I thought it's an interesting story because I think it's a real interesting story of the sort of sociology and history of science. Could be a nice New Yorker article if someone can find out how to write. But and I think that there was recently a New York article, not specifically about this, but about Alzheimer's disease in general. But what is the evidence? So there's been a big effort in gamma secretase inhibition, and I want to just talk about that for a second. So because I think there were some flaws in this pathway. But first of all, there's these rare cases, and a lot of them are in one family that's in northern Colombia, an extended family, 4,000 people in this extended family. That cause these rare autosomal dominant forms that are early onset Alzheimer's disease. And so there is a genetic link to that rare family, right? Whether it's relevant or not, we don't know. Now, of course, if that rare family was in New York or Japan or Israel, I think there should be drug development going on with that rare family. But the rare family is in northern Colombia, and so treating that disease, there's really that specific disease. So the question is, are they generalizable? Are the features generalizable? We know that presenilin, one of the things it does is it cuts APP to produce A beta. That's how it was discovered, right? It was named presenilin. There was all sorts of bias in it. It was named presenilin because it was connected to amyloid, because the first discovered function was the cleavage of APP to A beta. Now we know it does a heck of a lot of other things. And I always wondered, why would you think that the major function of this protein that's all over, expressed at high levels, would be to make a pathogenic protein? It just doesn't make sense. But the understanding of what the protein really did lagged, and that's a problem, as you see. The mutations, the first ones that people looked at, seemed to make more A beta, and so everyone was happy with that. But then when you look at now a list of 100, there's not a real pattern in terms of whether they make more A beta or less A beta, right? So the mutations, people thought that they were telling us something, that, oh gee, the mutations should make more A beta, that should make more amyloid, that should drive Alzheimer's. And maybe we should inhibit the formation of A beta that way. But that was, I think, flawed thinking. Nobody knows what else it does. Nobody knows that these features, that is, changed presenilin or changed A beta product, is not a general feature of sporadic Alzheimer's disease. Why are these? If the only importance of presenilin to Alzheimer's disease is in making A beta, why don't all the mutations that cause Alzheimer's disease do the same thing? They don't, with respect to A beta. So And one of these, right, is these guys, Ralph Dixon and colleagues reported 2 years ago at Cell that presenilin is required for autophagy, which is a very important cellular homeostasis event that gets rid of waste proteins and dysfunctional mitochondria and fat droplets and things like that in cells. So incredibly important process. And inhibiting this now seems to damage that process. So why would you, but people were assuming that the presenilin was a gain of function that they wanted to inhibit. I think the reason why people went into the clinic was because you could measure A beta in the CSF, and they realized that if they treat a person with a gamma secretase inhibitor, it should lower the amount of A beta in the CSF as a surrogate marker for the brain. It would be nice if there were an imaging marker for that event, but this is a surrogate marker for what's going on in the brain. And they developed really great technology to do this. This is a paper with David Holtzman and Randy Bateman at WashU. They have this great technology where they feed heavy atom amino acids, C13 amino acids, and then they measure the time required for that feeding to go through into APP, into A beta, and come out with labeled CSF A beta. So it's a mass spec technique at the end. So you see the C13 signal comes out in the A beta and then it goes away. And they could show this way, Lilly used this to show that their gamma secretase inhibitor actually reduced the rate of production of A beta by the brain. And they were so happy about that, I think they forgot to think about whether that would be good or bad, in my opinion. And I was saying this before all this. So what they found in so they went right from that trial. So a phase 2 trial is really your choice, right? Phase 2 is the choice of the developer. It's not an FDA, right? So here they chose to measure a marker they could measure quickly and make sure, okay, do we lower A beta? So this says yes, the drug gets into the brain, likely, and it inhibits that proteolysis event. And now this study, which was over $100 million, says that it has no effect on cognition. In fact, it makes cognition significantly worse, right? Which just blew my mind, right? Because I would argue that nobody thought about what else this enzyme is likely to do, this common enzyme is likely to do, because they were so reductionist, so focused on the one target. Not only does it do that, and certain friends of mine were telling me, well, this is because there was some known toxicity and it caused GI problems in patients and that can affect cognition. But they then brought the patients back 32 weeks after they stopped dosing and they remeasured cognition. And the drug group was still worse and by the same amount as they were. So basically they created a brain damaging agent in the gamma secretase. Yeah.
So it says here that, in that paper it says it's a potential treatment for Alzheimer's disease. So is this a specific APP gamma secretase inhibitor, like for a docking protein?
Well, I don't know that they really know that. I think it's an active site-directed inhibitor. They've done labeling in this case, gamma secretase inhibitors, right? And so now they have these gamma secretase modulators, which,
well, there have been 2 drugs. One was from Paul Greenberg's lab, and the other was from Neurology.
Yeah.
And they both said that they had APP specific.
Yeah, I would say Paul Greenberg stuff. I would, yeah.
But they both
wait till you see data coming from a pharma company. The one thing I will say about pharma is when they do data, they do it right.
But
they might do the wrong
Myriad
experiment.
the same thing. Myriad Genetics
Yeah.
published the same
Yeah,
thing.
Myriad's compound didn't even get in the brain. I didn't even want to talk about that. That was too depressing. They ran a phase 3 trial of a compound that they never demonstrated brain penetration. They had no exposure in brain and they spent $200 million. But someone paid them, I forget who paid them up front. Someone else took on the cost of that trial. J&J maybe, I can't remember. But yeah, that was too depressing to even talk about. But this is really interesting. So Bristol Myers then, of course, goes ahead with their trial. They have, and they find the exact same thing. Significant worsening of cognition. So what this has created is a problem in the investment. It's like, you told us, this was the best developed hypothesis for how to modulate the rate of progression of Alzheimer's disease. It was a complete and utter failure, and not even a failure, it went in the other direction, which was crazy. So I want to tell you just about 2 projects that I got involved in that, that have some of these problems to illustrate some of these points. So this is the project that I left academia for in like 2005. I spent 5 or 6 years at a company called Big Medicine to pursue this. I won't get into the reasons why we thought this would be true, but at the time I thought this was the greatest thing since sliced bread, right? What we found was that a known compound and a known class of compounds that were being developed for oncology called farnesyltransferase inhibitors actually could have efficacy, we thought, in neurodegeneration, and actually generally in neurodegeneration. And this is a compound that we licensed that was a Pfizer compound that went into the clinic for in a phase 1 study in cancer patients. These are patients who were very sick and had failed other therapy. And patients had been dosed with this without what cancer guys call bad side effects. But it's different, very different for cancer than it is for Alzheimer's disease, obviously, which I learned. And I actually went all over trying to find, so J&J had a compound called Zarnestra, which is a really close relative to that. In fact, it has a chlorine there instead of acetylene. And their chemists didn't write the patent well, so Pfizer jumped in with acetylene. But J&J had a compound in the clinic, and they ran probably 10 phase 2 trials in different cancers for this compound. I tried to get them to repurpose it. I said, just start a neurodegeneration program on the side. We'll do the work, They wouldn't do it, so I had to form this company. But so this compound, I thought it was a really great thing to have a compound that had already been dosed in man, and in the case of Zarnestra, a very close relative, dosed in man for up to a year at extremely high doses, 100-fold higher than the doses that we were working with. And I thought, this is it. It's a compound. It's a very clear and fast way to get into the clinic, right? And in fact, it wasn't, because a lot of the benefits, there was a little bit, people were a little less worried maybe about the risk of having toxin people because they had this analog to look at. But in fact, this was an old compound. And so the intellectual property for the compound itself expired at a time where we would barely have time to get 2 or 3 years of patent life in the clinic. And patenting something for a new use, people do that. We did it. It's not, people don't like, pharma doesn't like to do this. So in the end, actually, Link made their own compound that was an entirely new compound, and all of the value of the company, in the end, in the purchase by AstraZeneca, was judged to be the new compound, which hadn't even been in people, because it was chemically much more. It had 10 more years of patent life. The compound did great stuff. I mean, it was fun for a few years. It did really interesting stuff. Here's a really cool experiment that Anna Maria Cuervo at Einstein did, where we could show that this fusion of 2 vesicles that's critical in the autophagy pathway was accelerated by the compound. Did a lot of other really cool things. The problem is, how can you measure this in a human brain? It's a very fancy experiment, and we science guys all like to do the fancy experiments. But the fact is that a simple experiment that could measure that in the human brain would be really, really valuable. This is an imaging project, I think, right? But it can't be done by fluorescence imaging. It also had effects in the APP-PS1 mice. These mice, I think, are, they've been cured by 60 different strategies in the clinic, including secretase inhibitors. They overload the system with tons of A-beta, and you can cure it by reducing A-beta. That shouldn't be a surprise. The real question, though, is why is that relevant to the human disease? That it's overloading the system with a ton of protein when, in fact, people with Alzheimer's disease don't make a ton of it. They make about the same amount as people without Alzheimer's disease. But at any rate, we had nice data in mouse models of A-beta deposition, nice data in mouse models of tau deposition. Then we ran into a little bit of an issue, and that is that we had efficacy at a dose, and this is increasing dose in a mouse, we're measuring farnesyltransferase activity that remains in the brain. We had efficacy at doses where we weren't yet inhibiting the most of the farnesyltransferase. And that flew in the face of this target engagement idea, the idea that you have to occupy 50% of the target to get 50% of the effect. We got all the effect with none of the target, as far as we could measure, occupied. Did a lot of work to try to understand that, but we believe we're dealing with an isoform, so that there are probably multiple isoforms, and we're hitting one first, but we're never able to prove that. So where the link stood in this incredible limbo, which is we didn't have, even though we knew the target, we didn't know the mechanism flowing from the target with certainty. That creates a risk in their mind, because they say, gee, you know, I like to think we know the mechanism of our drugs because it makes me more comfortable. And then we didn't know how to measure anything in a human being that was relevant. All the effects that we showed in cell culture and in animals were cool, right? They were thought to be relevant to disease, but none of them could be easily translated into human. That, to me, that's the message of this talk has to be that measuring a drug response in a human brain, the biology of drug response in a human brain, is like really, really, really important. And that's the missing piece.
In the homology between a mouse or rat and farnesyltransferase.
Yeah, there was good homology. We made the human protein, and we had, you know, we matched up. That wasn't the
In
issue.
other words, KIs were.
Right. No.
What about other transferases like germ transferases?
Yeah, it was not. Yeah, no cross-reactivity. That's why we chose this one. The ones with cross-reactivity, I'm guessing you know, this got a lot of tox problems. Yeah. So
we're doing it for college.
Yeah, for oncology, yeah. So that project is still at AstraZeneca. We've been working with them a little bit to try to get that compound probably into Huntington's disease, because one thing is Huntington's disease is an orphan, and there's just a lot of other reasons to go after Huntington's disease. Let me just mention another project if I have time here, maybe 5 minutes.
You got 11 minutes.
Oh, I got 11 minutes. Okay. So this is a project I got newly involved in, and I got involved. I was aware of this, some of this science. Just got involved with a company called Lysosomal Therapeutics that is starting around this discovery. And I want to show this because, to me, this is like the ideal project in neurodegeneration. It's ideal, and it may not be. There aren't many of these, but this is one. And this came from a clinical observation. So patients who have Gaucher disease have very low glucocerebrosidase activity. They're treated by the Genzyme drug that they make over on Soldier Field Road. And that drug replaces the lost enzyme. And patients now, a lot of their symptoms are improved, and they're living much longer. They're living into their 50s and 60s. It's normally a childhood onset disease. But it was observed that a lot of them were getting Parkinson's disease at a much higher rate than one would expect based on their age. And at autopsy, these patients, their brains looked like typical Parkinson's disease. And the clinical presentation was very typical if you could pull it away from the Gaucher symptoms. What people did was then they took a look at the carriers of the Gaucher. So Gaucher is autosomal recessive. You need a mutation or a deletion or something on both chromosomes. But obviously the parents of those children turns out to have a higher risk. And now since in the last 5 or 6 years, there's been probably 10 or 12 really detailed, huge genetic studies looking at the frequency of mutations in glucocerebrosidase gene in Parkinson's disease. And they found that patients who carry the gene, who don't have Gaucher's, have about a 5 to 8-fold increased risk. Patients who are homozygous, who carry 2 bad alleles, have about a 20-fold risk, although that's probably a gross underestimate, I would guess, because, again, early Parkinson's is very difficult to diagnose, even in a healthy old person, not to mention an old person who has Gaucher on top of it. So high increased risk for 2 mutant alleles, moderate increased risk. Now, how common is this form? It's actually very common in the Ashkenazi Jewish population. So if you probably know when you have kids, they always ask if you have Ashkenazi heritage. So both my wife and I do, and we both got tested for that. And the reason is because they don't want to see 2 carriers. Well, they want to inform 2 carriers about the risks of having kids. So about 10 to 30% of Ashkenazi Jewish PD population carries this mutation, and probably 2 to 9% of the general population. The other thing about this that's just so great is that the Ashkenazi Jewish population is A, ethnically concentrated, and B, geographically concentrated. So you can run a trial in these people either in Tel Aviv or New York easily, right? Easily, and you get all these benefits of homogeneity that you don't get when you run a trial in 4 continents. You get all sorts of ethnicities, and we already know that ethnicity has a huge effect on neurodegeneration and progression of disease. We already know that from natural history studies. We're looking, and so this is just ideal, from a practical perspective.
Real quick question.
Yeah.
So the 20-fold increase in Parkinson's disease with the homozygous
Right.
gene patients, are they all early onset? 'Cause it says
Yeah, the onset is earlier than typical. I think I might have taken out that side of it. The onset is earlier than is typical for Parkinson's. So it's in the 50s somewhere. And then if you take a group of Parkinson's patients, a large group, and this has also been done, you separate the early onset from the typical onset patients and ask where are the GBA mutations now, the GD mutations, and they're preferentially in the early onset group. So even the carriers get earlier onset forms. And then there's an actual correlation between GD risk and PD risk that can be shown clearly in this one mutation, which is actually the most prevalent. The other thing about this, gene is that there are 300 mutations that have been identified in humans, and that sounds like automatically, oh my God, what a nightmare but 4 of them constitute about 95% of the population, and 1 of them constitutes about 50%, and in the Ashkenazi Jewish population, about 70%. So N370S is actually, you can work on it as a single mutation, right, in terms of clinical studies, which is fabulous, I think. What does glucose-3 residase do? It's a lysosomal enzyme, and Wikipedia says that its only function is cleaving glucosylceramide. There are no other identified functions. That's a glycolipid, so it cleaves glucose, the glucose head group, and it does so probably at the membrane surface or probably with a cofactor. So it's a complicated enzymatic reaction, actually. And the way the enzyme replacement works is it's just taken up by the endosomal pathway, and then it gets into the lysosome that way, and you're just replacing the lost material here. There's also a problem over here, though, it's postulated in the disease, and that is some of the mutations, or many of them, lead to instability in the protein and potentially slower folding, insofar as those 2 things are probably related. And that causes the presence of unfolded protein in the ER, which is a bad sign. It's a signal for what people call the unfolded protein response, or endoplasmic reticular stress. That would be a bad thing too. So it could be bad, but now enzyme replacement doesn't address this. It also doesn't penetrate, the enzyme is a protein, of course, it doesn't penetrate into the brain, right? So they can treat Gaucher with enzyme replacement, but they can't treat the risk that these patients have of Parkinson's disease with enzyme replacement. And they're experimenting, obviously, at Genzyme wildly trying to figure out how to get a protein into the brain. I don't think that's going to be an easy thing in next considerable amount of time. But at any rate, the deficiency leads, as I said, to unfolded protein here, buildup of substrate. And there are actually signals for this that occur in the CSF sort of acutely. You can see accumulation of substrate. Substrate levels in the CSF go way up. Protein levels, you can actually measure glucose-3 residase in the CSF. You can measure its activity. The total activity in the CSF goes down in the disease. So this is this really simple correlation where you have the rare lysosomal disease. And again, the nice thing about this is everybody likes starting with rare diseases now. Rare lysosomal disease, already been treated, right? Development path known, no questions about whether you can modify this disease, you can. And so these patients who have Gaucher have very low wild-type activity, very high substrate, and very high risk. And the patients who are carriers have moderate loss of GCase activity. We don't have data on this point.
So I'm not really
Moderate.
familiar with the ceramide and
Yeah.
dopaminergic neurons, but what's the connection there to dopamine?
Yeah, I don't know the answer to that. And I'm going to say right now, I mean, I have a lot of ideas, but one thing I've learned is that I have to suppress my ideas because we can think inside the box. It doesn't matter at some level. If there's a correlation between these two, I'm not going to worry about the pathway. Only because I'm not smart enough to work. You know, it just takes too much, you know. But you know, we have some ideas about that, right? And obviously, if you want to make this a risk-free, if you really want to comfort people in pharma, you would. But anyway, these two demonstrate that. Right. What we want, what the idea here is that should say activation. I don't know why that says inhibition, but the strategy then, of course, is to activate GCase using a small molecule chaperone. That's the strategy of the company that I'm working with. And what you see is there's these great clinical trial groups right away and markers that really avoid the problem that there are no good markers for Parkinson's disease, because you can use the markers for target engagement and then the surrogate, which would be substrate, for the downstream pharmacology. And you don't need to know what that pharmacology is, because you know that GCase deficiency is tied to disease in human beings. The genetics tells you. So you don't have to worry how that happens. I know that might make people uncomfortable. It used to really drive me nuts, but I'm fine with it. I'm fine with the ignorance. And then there is a class of Parkinson's disease of patients who are heterozygous. There might be 50,000 US. It's a perfect size for a orphan disease, where you can treat them, and in a phase 2 trial, you can just demonstrate that you can boost GCase activity in their brain, and that really should be essentially a surrogate for efficacy. You don't have to look at PD signals in that trial. So once you know that, then you can then justify the expense to run an 18-month trial to clinical proof of concept. Because, of course, in order to get it approved for Parkinson's disease, you have to demonstrate that there are tangible effects, there are tangible things that can be measured, improvements in a patient's life that can be measured. So maybe not even some composite rating score, but maybe number of falls per month, or some gait measurement, gait speed, or balance, something like that, that really tangibly affects the life of the patient. Those are hard measures to make, require a lot of patience. And again, in order to get to that point, you have to demonstrate in an early trial that your drug is doing what you think it's doing in the brain. Now, here's an imaging project that I'd love to get. I'm trying to get a lot of different people interested in this. I think you can easily image for glucocerebrosidase activity in the brain. There are affinity reagents for the compound that have been used in different ways, but I think imaging brain GCase activity would be preferable to imaging CSF GCase activity, because the CSF is not the same as the brain, and it would just be really nice to be able to do that in brain. So that's sort of the message that I wanted to leave you with. I think this. Will there be others like this? I actually think there will. I think the interesting thing is in these rare recessive diseases, there are a lot of carriers out there, and a lot of times the carriers don't have the same sort of clinical presentation as their children. But some of these questions about late onset diseases and about risk haven't really been asked in this. In this, the only reason they ask this question is because the clinical observation that the Gaucher clinicians observed this and wondered about this. I mean, that's so old school, right? There was no GWAS that led people to, in fact, they missed it. So somehow, it's a clinical observation that led to this. And I think there's going to be a lot more of those. So, given the current situation economically, I think the only way into Alzheimer's and Parkinson's is to divide it up, to divide it into pieces that are tractable, where you can really understand the patients, where you can run a phase 2 trial with a homogeneous group of patients and show they clearly respond to the drug in the intended way. This is what Myriad never did with their gamma secretase inhibitor. They never knew that it ended up not being in the brain. And then go forward from there. So, anyway, that's all I have.
You made the comment that the enzyme doesn't go into the brain.
Right.
We know it gets into cells, right?
Right.
Do we know for sure that some of those cells don't get transported across the blood-brain barrier? I mean, maybe this points to your imaging experiment, is that, do we have a way of detecting it? And then what is the level, the amount that needs to get across?
The reason I said there were affinity reagents for the active site, they actually used this at Genzyme. They made a fluorinated active site suicide substrate, labeled the active site of the enzyme, and then did an infusion and did a biodistribution study. But actually, I'm sorry, this was probably done in mice, and they didn't detect drug in the brain that way. But yeah, I'm not, obviously not privy to what's going on there, and I'm just guessing from their crazy, their affinity now for gene therapy methods and viral infection methods, shunting, and all that stuff that they really, need to get enzyme into brain. And also, a protein drug is never. I mean, people like protein drugs, but if you could replace it, a small molecule would be much preferred. That's what's happening in MS now. You're seeing all these small molecules are going to, I think, blow away the big molecules, just because getting an infusion every 2 weeks, which is what Gaucher patients have to do, is a real hassle. And they'd much rather just take a pill every morning. So I think that this will move in the direction of small molecules. Gaucher. Yeah.
In terms of Alzheimer's disease being disaggregated into different diseases, there is a gene which unfortunately I don't remember. It's a gene that for a protein that handles RNA, binds to RNA, and it's related also to ALS, but this is a form that is the frontotemporal area of the brain.
Yeah.
And that does give Alzheimer's-like symptoms.
Yes. Yeah.
We know
That's
this.
TDP-43?
Yeah, that's it.
Yeah. Ironic that you forgot that.
What?
That you forgot the name of that gene.
Yeah.
Anyway, maybe it's coincidental.
Well, since I deal with tons of different therapeutic areas, that's why I'm not just.
Yeah, no. I understand. I think, that's another potential lead. The problem is seeing how that's druggable. It's not,
It's probably not the target that's directly druggable.
Yeah.
Anyway.
Yeah, that's the case. I think that's the case with all the deposition diseases, that the druggable target is not the deposition itself, but something either upstream or downstream of the deposition. Or it's possible the deposition is a red herring, which is something that.
And one of the early days of learning about that gene and that disease, where sometimes in the brain, sometimes in the motor neurons.
Yeah.
So in terms of understanding the pathway, we're not there yet.
Oh, yeah. Well, no, but, I know that argument. I mean, it's a clear argument, which is science. We need more funding for basic research because we don't know as much about it.
Well, they're doing the basic research.
That's true. It's true. It's a true statement, but I think that argument has to be made to NIH also. That is, that we don't know, that is, I think that the understanding of these diseases is still really early on. And so we don't have attractive targets in most of these cases. Gaucher disease is a very simple disease, right? Very simple, but Alzheimer's is very, very complicated. So it may be that it's, but, that's really talking about time.
No, but in this case, we actually had funding for doing the basic research, but still I don't think we're there yet on the target or on the animal model, which is why I was interested in it. Yeah. I think, like I said, I just think you could bottle drugs that were ready for human use for Alzheimer's disease, and you still would not get investment. That's the way I feel right now, is that it's not a question, it is, there aren't enough good targets, and there aren't enough good compounds, but that's not the problem, I don't think, because I think even if you had them,
people wouldn't know how to do the first human experiment. So they wouldn't even try because the investment required to do that. That's an opinion. I think it might be changing now because, you know, the good thing is now the IPO market is opening up and venture capitalists can get their money back in different ways than selling it to pharma. So, you know, maybe
That's
that'll
Peter, so.
Rare disease.
Over
Just-
rare
Do
disease,
you have that same connection?
Yeah,
No.
yeah,
Someone that,
yeah. I mean, you know, I think,
Oh, James.
You commented that there seemed to be a disconnect between.
Okay, so we can take this out.
filling the
Exactly.
that line of division relative to the biological.
right,
Then
right.
you said what's going on in testing other diseases.
Yeah, it's a BGA too.
So,
Same thing. Yeah.
I mean, this is a case where we have a lot of evidence that, something that I can't share with you. Things that,
There, the two,
Oh,
right
so
there.
this is-
Yep, same.
that
Thank you.
there's a subclass.
That was
Thank you.
going up.
So there
Okay,
is
thank you.
strong evidence that there is farnesyl transferase inhibition, but it's a subclass of the total farnesyl transferase. So it's swamped out by, you know, let's say 99% of brain farnesyl transferase is not inhibited by this. 1% is. We have a lot of evidence.
Okay.
See what I mean? So there's a subtype, and that type is producing efficacy. This type is irrelevant. When we measure whole brain, we get,
do I need to go to preferences and
And then you don't
select
currently have
all?
a way to measure that.
we don't have any way to measure target.
Looks like it unplugged on you.
Let's, so catch up with.
Yeah.
There you go. They just
Great. I
unplugged
really
it.
appreciate it. Great talk. I'll introduce Alice while she's getting her talk up here. Alice is from Caltech, and she's going to talk to us about Drosophila as a model system. I'm actually quite intrigued